Protein synthesis in eukaryotes. First stage is called transcription. Firstly, the DNA is opened by the enzyme DNA helicase. The promoter region, known as the tartar box, is identified by the enzyme RNA polymerase, which then binds to the promoter. The DNA code on the template strand, otherwise known as the antisense strand, is the only strand used. The RNA polymerase reads the template strand from 3' to 5', making a complementary pre-mRNA strand. It uses the free-floating nucleotides in the nucleoplasm. It installs uracil instead of thiamine, and the mRNA made runs 5' to 3' and is single-stranded. The pre-mRNA breaks free when the termination sequence is reached. A modified guanine 5' cap is put at the 5' end. A poly-A tail of about 250 adenines is attached to the 3' end. Both the 5' cap and the poly-A tail stabilize the pre-mRNA. The pre-mRNA undergoes splicing at the spliceosome. The introns are removed, leaving only the exons. This makes mature mRNA. The mature mRNA then leaves the nucleus through the nuclear pores. The second stage is called translation. Once in the cytoplasm, the mRNA moves to the ribosome. The ribosome is composed of 2 subunits of rRNA. There is a groove between these 2 subunits. The first codon enters the ribosome and binds to the P site. tRNA molecules approach the ribosome, and the one with the correct anticodon drops off its amino acid. Hydrogen bonds form between the matching codon-anticodon complex. When amino acids are dropped off, peptide bonds form between the adjacent amino acids. The tRNA, once it's released the amino acid, moves into the cytoplasm to pick up exactly the same amino acid. This continues until the stop codon is reached. The polypeptide breaks free and undergoes folding. The primary structure of the protein is the amino acid sequence. The secondary structure is the spirals and pleats formed. The tertiary structure is whether the polypeptide is globular or linear, and the quaternary structure is whether the polypeptide requires another polypeptide to be fully functional.